Okay, so I think we have both audio and video. For those of you who aren't in the room, I'm Steve Tillis, and Len just met Sharon today. And I've known Sharon for a while, so I was going to introduce her. So we're really fortunate to have Sharon here from Stanford, a associate professor there and director of the Sean Parker Institute, PI at CoFAR, the National Institute of Health Consortium for Food Allergy, which has paved the way for a lot of our thinking in the space. And today she's going to talk about new therapies, as you probably heard of Omalizumab, and also some others. So we'll give the floor to Sharon. Thanks.
Thank you, Steve. Thank you, Steve, and thank you, Len, for the kind invitation. I'm excited to talk to you about emerging therapies in the food allergy space. And so please feel free to interrupt with questions as we go along. Let's see. These are my disclosures. I advise for a number of different industry partners in the food allergy space. So here we are in 2024. I started my career at Stanford straight from fellowship 11 years ago. And when I started, there was no therapy besides avoidance for food allergy. And in 11 years, we now have 2 FDA-approved therapeutics for our food allergy patients. So it's really amazing to see the progress and how food allergy researchers have leveraged the work that's being done in other allergic disease spaces to apply those biologics, therapeutics for food allergy disease state. So Palforzia is peanut oral immunotherapy product. I'm sure you all know well, approved in 2020 for peanut allergic children. And Omalizumab is an anti-IgE biologic, just recently approved in February for treatment of food allergic children and adults. And I'll go through some of that data, focusing on Omalizumab and other biologics. So I think I don't have to tell everybody in the room how Omalizumab works, but this is a nice recent review by our colleague Tom Casale and Matt Greenhot and Alessandra Fiocchi, looking at Omalizumab mechanism of action, and actually reviewing where Omalizumab hits the target for food allergy and other questions that still remain. But as we know, Omalizumab is a biologic that reduces free IgE in circulation. This reduces the cell-bound IgE on mast cells and basophils, which then reduces the high affinity receptor, the Fc epsilon R1. We have reduced allergen binding and cross-linking of IgE on mast cells and basophils, reduced mediator release, and reduced allergic reactions. We all know what the symptoms of allergic reactions that occur in IgE-mediated food allergy, which typically occurs within 30 minutes to 2 hours following an ingestion of the culprit food. And we hypothesize that anti-IgE can really eliminate a lot of those reactions if we have good reduction of circulating IgE, and this pathway is effective. So I'll talk a little bit about the OUtMATCH study. It was recently published in the New England Journal of Medicine from the Consortia for Food Allergy Research Group. And this is just stage 1 of the 3 stages of the OUtMATCH study. And I'll talk us through it. So Stage 1 was designed to investigate omalizumab versus placebo injections for 16 weeks in multi-food allergic individuals. So 435 individuals from age 1 to 56 were screened. There were a number of individuals that were screen failed. They had to have an allergy to peanut plus 2 other foods. Those foods were a choice between 6 other foods, including milk, cashew, walnut, hazelnut, wheat, and the 6th one is placebo. So everybody had to undergo at least 3 food challenges at screening to make sure that they were allergic enough. So a number of individuals failed at the screening food challenges. A number of individuals failed because their IgEs were too high. And that's not really accurately reflected in the consort diagram because a lot of the 10 sites already had IgE levels on patients that were coming in and knew that they weren't going to meet the entry criteria. So in reality, there were a lot more patients that had higher IgE levels than what's in the paper. So ultimately 258 were excluded, and then 177, actually 180 adults and children, 177 were just children, so 3 adults, who qualified with entry-level reactions for peanut plus 2 other foods, were then randomized 2-to-1 to receive omalizumab versus placebo for 16 to 20 weeks. And then from them, then they did another set of double-blind placebo-controlled food challenges, and 59 of those individuals went on to an open-label extension of omalizumab for an additional 24 weeks. And this was something that the FDA asked us to do to see about the durability of the effect in this patient population. So this is the consort diagram. Again, after stage 1, which was roughly 16 to 20 weeks, then patients went on to open label extension. This was at 10 sites across the country, aiming to get good geographic distribution here. So our demographics were a slight skew towards male children, 57%. It was fairly nicely split across the different age groups. So when we're breaking down the children into 1 to 5, 6 to 11, and 12 and older, a pretty nice representation across all those age groups. And we had predominantly Caucasian race, which I think is one of the limitations that we had less than 8% African-American patients who were enrolled in this study. And I think we all strive to include diverse patient populations, but it's harder to engage research participants in, that take time-consuming activities, including all-day food challenges in the clinic. This is the omalizumab dosing table that was used in this study, and this was based on phase 2 studies that we had done with Steve Tillis and his group, our MTAC study, which we leveraged for the Outmatch study as well. You can see that the IgE cutoff is 1850. But, if you're a larger child or adult, that IgE cutoff is less than that. And the max dosing was 600 milligrams. And these were blinded injection pens.
The same as used for asthma?
Yes, based on the IgE weight base, but extended.
Yeah, because it stops at 700 for asthma?
Yes. So what we found was that omalizumab works for food allergy patients across peanut and other foods. And this is looking at the peanut allergic individuals. And then we had nice representation across the other 6 foods that were studied: egg, milk, cashew, walnut, hazelnut, and wheat. And we had some small placebo reactors or responders, but very nice response. And what I'm showing you is the different doses at the food challenge on the x-axis. And so you could see that about more than 50%, 2/3 actually, were able to pass a peanut food challenge after 16 weeks, up to 6,000 milligrams of peanut protein cumulatively at the end of the food challenge. One thing to note is that our entry criteria for peanut versus the other foods was very strict. They had to react at 100 milligrams or less. And our exit criteria was 2 levels higher than that. So 600 milligrams was the primary endpoint. So could peanut allergic patients that were highly sensitive pass a food challenge to 600 milligrams after 16 weeks? Thinking that this was the precedent that was set by Palforzia for regulatory approval. And in reality, it's about 2 to 3 peanuts. So an amount or an exposure that is relevant for accidental exposures. For the other foods, the entry criteria was a little bit higher. It was 300 milligrams. And so the exit criteria was 2 steps higher than that, which was 1,000 milligrams. And the reason it was designed with these different cutoffs was because we wanted to include as many patients as we could. And so we were hoping to have less screen fails at the food challenge entry time point. And then in retrospect, most of these individuals who qualified were all extremely allergic to, all 3 foods. I think the median dose that they reacted to at screening was, the 30 milligram dose across all the foods. And so the threshold to be considered successful for the other foods was 1,000 milligrams.
Yeah, sorry to interrupt. But,
Yeah.
the point of confusion has always been, was the highest tolerated dose or a listing dose? So the dose at which they had symptoms that they couldn't keep going, or the last dose they tolerated before they
That's a great question. So it is the highest dose that they tolerated.
so the closer to the immune way than the
Yeah.
DTV way for okay.
Yes. So put it in perspective of food equivalents. So 67% could ingest 600 milligrams without dose limiting symptoms. So cumulative tolerated dose, and which is roughly about 4 peanuts. And then 57% could ingest 4,000, about a tablespoon of peanut butter. For those patients who were randomized to the Omalizumab arm. For egg, we had a slightly higher success rate down the line, similarly for milk. For cashew, our success was a little bit lower. Only 41% were able to tolerate 6,000 milligrams, the last dose tested at the 16 weeks food challenge. But those cashew allergic individuals also were extremely allergic at the screening food challenge. And so that was an interesting thing that we saw across all the different allergens. When we look at the success rates across 1 food, 2 foods, 3 foods, based on the cutoff of 600 milligrams for peanut and 1,000 milligrams for the 2 other foods, we see that 1 food has good success rate, 2 foods has a little less, 3 foods has a little less than that. And that is similar to what we saw in our phase 2 studies with MTAX and with MapX, that when you add more allergens to the mix, your success at certain thresholds diminishes. But if you increase the time on drug, or the different thresholds, lower thresholds, you have different success rates. So it's important to kind of understand where the cutoffs are for duration of therapy and the dose that we're considering successful. Yes.
When this drug first came on the market 20 years ago for asthma, we were told to try and get the total IgE under 10, although we never had a way of measuring it because clinical available. Did you measure total IgE? Did you go for the same goal?
That's a great question, Len. So we partnered with the company, and yes, there is measurements of free IgE at that 16-week time point. And most people were below the 15 cutoff point. So suggesting that this dosing table that we borrowed from asthma has a similar effect in our food allergic patients. So it's a very safe drug. It's similar side effects that we've seen in our asthma and urticaria patient population. Most patients had injection site reactions. A few had hives, a few had hypersensitivity reactions, but nothing that was unexpected. So in conclusion, for the Outmatch study, we see that 2/3 of food allergic individuals had improved threshold sensitivities to peanut and other foods after 16 weeks of injections. And some of these patients received injections every 2 weeks or every 4 weeks. Omalizumab's effect on cashew allergic patients was a little bit lower than we were expecting, but in that open label extension where they received additional 24 weeks, we did see the success rate climb for our cashew allergic patients. So for cashew in particular, they need a little bit longer time on study drug. So this has now led to the FDA approval of omalizumab for food allergy, this plus the phase 1 and phase 2 studies. And the indication is pretty broad. So we were very excited about this. It's indicated for IgE-mediated food allergy in adult and pediatric patients as young as 1. So the Outmatch study was the first to study omalizumab in the 1 to 5-year-old age range. And it was safe, it was tolerable. It's meant to reduce allergic reactions, including anaphylaxis, that can occur with accidental exposure to one or more foods. This is to be used in conjunction with food allergen avoidance. And I don't know if anybody's prescribed omalizumab for food allergy patients here. Yes?
Yes.
With good success, approved.
The insurers in Washington State are taking their time approving it. I haven't defined criteria, but I've tried in a few patients. I'm still waiting for that.
Okay, great. And go ahead.
Has extended Genentech's patent life on the drug to have a new indication?
I don't know. I don't know that they know. Yeah, Karen?
It hasn't.
It has not.
Yeah, my understanding is that the biosimilar is still around the corner. It's been around the corner for a long time.
Yes.
There isn't anybody else making this product, is there? Even though it's off patent.
Well, I mean, the only pathway is a biosimilar.
Yeah.
There are some in development, I think, but they're not close as far as I know.
Yeah. So clinical context for this, it's omalizumab, as you all know, is given as an injection subcutaneously every 2 to 4 weeks, depending on that IgE dosing table. It has comparatively fast acting, but maybe not as fast as some of our patients or families would like. It has the advantage of being able to protect against multiple allergens, as well as influence other allergic disorders. And other therapeutic options like OIT may offer specific protection, but may have higher treatment burden. And I don't want to underestimate or undervalue the treatment burden of injections. So there's a lot of managing expectations for our patients and our families. And then all the tips and tricks that we've known for allergy shots about minimizing injection site pain and redness, including Buzzy Bees, ice, lidocaine creams, rewards and incentives, which we all do for all of our pediatric patients and adult patients. So I put this slide in there. I mean, this is no surprise for you all, but I think it's important to remind our patients and families that omalizumab is an injection, epinephrine auto-injectors are injections, but these are two very different drugs. And I think this is a question that comes up from our food allergy families. You know, do I need to carry my epinephrine auto-injector? Now I'm getting these injections. And just to reiterate and re-educate, that epinephrine is immediately a short-acting drug that is given after exposure. And I advise all my patients that they should still be carrying epinephrine auto-injectors. So some of our limitations with omalizumab, it protects against food allergy, but it's not curative. You know, we are certainly studying this longer to understand longer duration of treatment and whether there's durable effects from there. The protection varies across individuals, and at the moment, we don't have a great predictor of who are the best responders and who are the least responsive. There's different degrees of protection in individuals across the 3 foods that they were allergic to. So for some foods, there's a nice about 20% that were able to tolerate the top dose, 6 grams of all 3 foods. But there were also 14% that were not able to tolerate even 30 milligrams at the exit food challenge, right? And so we, you know, and then there's people in between. That is the $1 million question.
Does it get better after like a year or 2?
Only time will tell. That is right. And so and food allergy is a chronic disease, and it will require long treatment courses. But we don't also know if we have the potential of disease modifying effect in this 1 to 5-year-old patient population. We certainly see with oral immunotherapy and patch that there seems to be better responses in the very young toddler group, like 1 to 2-year-olds, and better durability of treatment effect when we're looking at sustained unresponsiveness testing and SLIT and OIT in the 1 to 2-year-olds. And so could omalizumab be disease modifying in a particular age range? You know, I don't think we know the answer to that. But if we look at allergic asthma and 6 years and older, we know that when we stop omalizumab, allergic disease comes back and asthma returns. So our guess is that it's going to be similar for food allergy.
A simple question, maybe you answered it already. When the FDA approved it, do people have to be multiply food allergic, just like the study, or can you give it in just peanut allergy?
You could give it just for one food.
Just sesame if you want.
Yeah.
Or whatever.
Or
You can
chickpea.
even give it for any food.
For any food.
Even
Yeah.
those that weren't in the study.
Yeah. Yeah.
How about the cost, and how does insurance cover it now?
That is a great question. So it is a costly drug. For those patients that I've had it approved, they're just, you know, they have the copay for the injections. Is that right? But they haven't had a ton of out-of-pocket expense. I don't know. Yes.
This is just a matter of courtesy. Do you think you could repeat the question into the microphone so that when we record it later, we know what the original question is?
Oh, yeah, sure.
Thank you.
What about the cost to the patients for Omalizumab? I don't know. So I will tell you, in Northern California, we have different payer systems, and Kaiser is one of the most strict healthcare systems, and we have Kaiser patients who've had it approved and covered. So there's very minimal cost to the patients there, and that's what I've seen across our Stanford patients as well. But and that includes like Blue Cross.
Yeah, I heard a rumor that some of the Blue Cross plans are sticking to the protocol. In other words, it's less than 3, they're pushing back.
Oh, really?
So, I mean, I don't know how that's gonna work,
What
but,
do you mean
but
less than
be
3?
prepared
Less
to
than 3
require peer-to-peer discussion.
How do you mean less than 3?
Well, the protocol required you had 3 allergen
Yeah, that's,
that you're, but the label, it could be just 1.
Yeah.
And so if, if it's just milk or something, it may be much harder to get it approved depending on the patient's history.
well, it's just like the trick that the insurance companies did when they got approved for urticaria.
Yeah.
Urticaria, they just had to be on a single conventional dose of antihistamine. Once that was proven, then they wanted quadruple dose of antihistamine plus an H2 plus leukotriene inhibitor. All of those things failed. So they made the criteria for approval much harder than the study indicated in the first place.
Yeah, no, it's an interesting story. Yeah, they're gonna do whatever they can, especially
Yeah.
if it succeeds. In other words, if it starts to be prescribed like crazy, then all of a sudden things will tighten up.
Yeah. I have had some scenarios where patients are on oral immunotherapy for one food and then want now to start Xolair for their food allergies, and insurance companies have pushed back saying that this is supposed to be used with allergen avoidance. So you can't use it in conjunction.
Well, that's an interesting.
So there's going to be
Pay for any of the oral immunotherapy.
Yeah.
So it wasn't.
So we'll see. I mean, I think these are the early days of trying to use this for our patients, and I'm sure we'll all have lots of hours with our peer-to-peer. Yes.
Can I ask, given what you talked about in the patients, just the types of patients who were included, and if there were only 3 adults in that cohort, what are your thoughts on use of Omalizumab for this indication in adults?
Repeat the question.
Yes. So the question is, what about Omalizumab for adults? Because there were only 3 adults included in this study. The phase 2 MTACs, I think, included a few adults, and we've had adults at Stanford who've been treated with Omalizumab. It's interesting because oftentimes it's the other way around. You get an approval in adults and then you extrapolate to peds. In this case, actually, because of the great amount of information, at least for safety wise, for Omalizumab in adults, the argument was made to extrapolate from the peds data to the adults, and they were included in the label indication. In our hands, we see good responses for desensitization in children and adults. And looking mechanistically, we don't see any differences in the immune responses for antigen-specific desensitization, which makes me think that the mechanisms of allergy in children and adults are very similar. So I think that Omalizumab will work for adults as well. But again, I think we need more data to be able to say that confidently, because we don't know what the range of response will be in adults, and will that be different? When I showed you those 3 different age groups for the children, it was equally effective in all 3 age groups. So that was also very interesting, because that's not what we've seen for SLIT or OIT, which is nice, because it's a therapy that doesn't have an urgency, that our families feel for effectiveness.
And isn't one of the strongest arguments for it to protect a young adult while they're in college? I mean, because even if you don't think of it as a lifelong therapy, it seems to be a particularly high risk time with cofactors and everything else. So the fact that there were few in this study, I think they got the benefit of the doubt from the FDA based on thousands and thousands of other adults that have been dosed with the same, time will tell.
Time will tell. I mean, I think this Steve's point is an excellent one. This is ideal for young adults who are going away from home, going to college, starting a new job, traveling, and all of these things that our food allergy patients have so many restrictions in their social life, and it affects the way that the jobs that they choose, and the different activities that they choose to engage in. So I think that there are certain adults that I think will really gravitate towards this. And then there's, other adults who say, hey, I've done a great job managing my food allergies up until now, and I've had very few accidental exposures, and I'm used to this lifestyle. And I don't know necessarily that they may feel that they don't need to address this, but I think this is where all of those conversations are long conversations with our patients that I think go beyond our 15-minute time slots of risks and benefits. So the big question is how, as we start this in practice, do we monitor response? And there's not a great test. These are some of the diagnostics that we have for our food allergy patient skin prick test, allergen-specific IgE, and then bead-based epitope assays. Those are all commercially available. Then we have mast cell activation tests and basal activation tests, which are more research-based tests, not widely available. And then we have our oral food challenge. And so really, the best test that we have is the oral food challenge. And it's not a slam dunk because there's so much that goes into it. What is the wait time here for a food challenge?
3 to 6 months.
3 to 6 months. Yeah. And that's everywhere, so I think that the infrastructure to be able to do this is challenging. And so with time limitations, are people going to be doing food challenges before they start patients on therapy? What are the minimal amount of tests that you're going to do before you start someone on omalizumab? Because once you start, free IgE is not a clinically available test. And if you try to check specific IgEs, you may have falsely high because you're capturing omalizumab bound IgE as well. So there's a lot of opportunity for further development of some of these tests or better markers. Yeah.
Have you looked at taking the ratio of the food-specific IgE to total IgE before and after Zola and see if there's any value in there? Because you would expect it to go down on Zola, right? So I'm just curious if the ratio stays stable, if that changes, if there's any diagnostic value.
That's a great question. So we did not. Oh yes, sorry. What about specific IgE to total IgE before and on treatment with omalizumab? Is there any marker there to understand disease responsiveness? So we only looked at total free IgE. We didn't look at allergen-specific free IgE. But when we look at specific IgE to total IgE ratios at baseline, that was not predictive of treatment response at 16 weeks. But
my understanding is, you think about it, without the allergen, I'm not sure why that ratio would change. Whereas with OIT or SLIT or whatever, there's this very predictable pattern of the specific IgE over time, increasing short term and then coming down, and then eventually over several years going way down. So but I'm not sure why blocking IgE without using allergen would change that relative thing. So like hypothesis would be that it doesn't, but it'd be great if it did.
Why'd you ask the question?
Well, I trained at Virginia and alpha gal, the ratio is pretty important for predicting whether they're gonna pass or fail food challenge because those reactions are delayed. That most people don't do the 6 to 8 hour challenge. And so
Yeah.
the ratio is pretty helpful. And I was just curious, is there any value there? And looking at that ratio, because I kind of use it as a marker when I see my food allergic patients, more not based on any hard published data on what happened, but I was just curious because I mean, if you test a kid with eczema, there's a bunch of low positive IgEs, right? And a very high total IgE. And so I consider the ratio as one data point when making a decision about the pretest probability of food allergies.
Those are all great
Oh,
points.
not the effect of the treatment on the baseline.
The baseline?
Yeah, you're not following it like a year later, seeing effect.
So I always check a total IgE with whenever I check specific IgE for any food, and I do like to track that ratio over time to see if it changes.
Yeah. So lots of open questions for young physicians and patients and our physicians who treat food allergy patients. And really, I think this kind of data lends itself to be captured in a registry, which we've started to do at Stanford. But so how long do we treat for? What about patients? This is the number one question I get. What about patients with higher IgE than what's on the dosing table? Greater than 1850 at that very lowest weight range, but less than that at the higher weights. Can this be used in combination with other therapies? Will we be able to introduce real foods into the diet? What markers can be used to predict response to omalizumab? And what would a better therapy look like? And so some of these questions are under investigation, and some of these therapies, especially looking at combination effects, are things that have been studied in phase 1 and phase 2 studies for quite a while, and will be studied in the Outmatch study as well. So for combination therapies, I thought I'd put a slide up. This is similar to the MTAC study. So Steve, this should look familiar. This was really work pioneered by Kari Nato at Stanford, and Tina Cinder, another co-investigator at Stanford, has done a lot of this work, especially in the multi-allergen space. But the concept here is that you treat with omalizumab for 8 weeks alone, really trying to drive down free IgE. And then you introduce antigen-specific therapy with oral immunotherapy. And in our phase 1, phase 2 studies, we've seen that you can do multi-allergen oral immunotherapy at the same time, so not sequentially, and that the rate of increase can be faster under the protection of omalizumab because you've taken out IgE, specific IgE out of circulation. And so the purple is what the escalation doses or the ramp of desensitization with allergen-specific therapy with Xolair versus without Xolair, which would be a slower pace. And then oftentimes patients or these protocols look at a maintenance period where you maintain allergen-specific immunotherapy, and then you could look at sustained unresponsiveness testing. So the omalizumab is 8 weeks before you introduce foods, another 8 weeks while you introduce foods, then you stop omalizumab and continue on with the buildup or maintenance of oral immunotherapy or allergen-specific immunotherapy. And we've seen that that's an effective way to use this in combination. We've looked at the asthma-based dosing with the IgE and weight-based dosing of omalizumab. We've also looked at this in the urticaria-based dosing for omalizumab with just fixed injections over 8 weeks, and then introduction of food. And both approaches have been effective in small phase 2 studies. So Outmatch is studying this in a larger patient population. This is what stage 2 of Outmatch is. It is everybody who started in stage 1. The first 60 patients went on to open label extension. The rest, 120, approximately 120, go into stage 2, where they're randomized, re-randomized to go into omalizumab facilitated multi-allergen oral immunotherapy versus omalizumab with placebo OIT. So in practice, it's omalizumab alone versus omalizumab facilitated multi-allergen oral immunotherapy for a year. Those patients will then have food challenges at the end. And then stage 3 is looking at dietary food introduction and with real food equivalents. So converting oral immunotherapy into real food. So those are still on the horizon. I'll switch gears really quickly to talk about other therapeutics that have been studied and are underway. So dupilumab, anti-IL-4 receptor alpha antibody, has been approved for asthma, eczema, and EoE, and studied in food allergy with a good theory for dialing down type 2 inflammation and really targeting IL-4, which is necessary for the production of IgE. And there's some data, small data, particularly from the atopic dermatitis studies that, were influencing epithelial barrier. And so really getting into the mechanisms of how does food allergy begin, dual allergen hypothesis, are you getting exposed through deficient skin barrier? And if we heal the skin barrier, can we influence the development of food allergy sensitization and food allergy? So dupilumab has been shown to reduce inflammation and repair epithelial barrier integrity. It's also shown to reduce opportunistic bacteria and increase biodiversity. Omalizumab has small studies have shown that there's changes in beta diversity of the gut microbiota. And so these are other ways that biologics may influence our immune system that can then lead to the sum effect of what we're seeing in our food allergy patients. So there's a lot more to try and understand the mechanisms that the target mechanisms that we're seeing beyond IgE for these biologics that may influence clinical outcomes. So the dupilumab studies, there were 2 that were sponsored by the company. They are not published yet. They're still in review. There is the dupilumab monotherapy study, which I won't talk about. And then there was dupilumab with AR101. That's not been published yet either, but I'll show you some data that's available publicly on clinicaltrials.gov. But before that, the AD studies, and this is a nice study by Speakerhoff, have shown that dupilumab can decrease specific IgE over time in patients with atopic dermatitis. And they looked at actually food-specific IgEs and a whole panel of them. I'm just showing you peanut and hazelnut. And this is over 156 weeks. The dupilumab studies really targeted their primary endpoint very early at the 24, 26 week time point. So this is some of the data that's publicly available on clinicaltrials.gov of dupilumab in peanut allergic patients used in conjunction with AR101. So it was a phase 2 study looking at the combination approach in almost 150 peanut allergic children. Similar demographics to what we're used to seeing in our food allergy studies. And patients were randomized 2-to-1 to receive dupilumab plus oral immunotherapy or placebo, dupilumab plus oral immunotherapy during a 28 to 40 week, during a 28-week updosing period, and then maintaining to 40 weeks. And then the primary endpoint was desensitization at the week 28 time point. They published on clinicaltrials.gov that there was a benefit for desensitization with this combination approach in the arm that received dupilumab plus AR101. So 56% were able to pass the food challenge to 2 grams cumulative peanut protein compared to 36% in the placebo group. It's a relatively well-tolerated drug with less AEs in those patients who had dupilumab as an adjunct therapy to peanut oral immunotherapy. So it'll be interesting to see where people take this. You know, it's approved in children as young as 6 months of age, and that age change happened after the study was already underway. And so we're going to have a lot of infants who've been started on dupilumab, and the question in my mind is, do we change sensitization patterns? You know, are we able to influence secondary prevention of food allergy in a highly allergic patient population who've already declared themselves as allergic at 6 months with AD?
So, okay, that's what I was gonna ask. So these kids are put on it for AD.
Yeah.
And you want to see if it reduces new sensitization early in life.
Yeah, that would be my question. I don't think that that's a study underway, but that's, you know, something again where I think, you know, we could look in our clinical practice.
But the company's not pursuing that.
No.
But they have a different combination.
Yeah.
Wipes out B cells and then that's with dupilumab.
Yes. Following.
I think that's
Yeah.
So this is part of the problem. The barriers to innovation is that it takes so much money and time to do these studies that,
Yeah.
that they have to make decisions that aren't necessarily, you know, for us the most exciting path, because that is the obvious. I mean, I bet it would work.
Yeah.
I mean, it just makes sense.
Yeah. But like you said, I mean, to enroll a sufficient patient numbers to be able to see this takes a lot of resources. And in when it's already out in the market, you know, I think this is where some of it is left to the practicing allergists to kind of sort these things out and see, you know, which is the best choice for their patients.
But I would say potentially an urgent issue in Seattle and probably many other places is figuring out logistics and clinics for seeing doing more challenges in little kids, because you can't wait a year if they're 2 years old. You can't, but the cat may be out of the bag, right?
Yeah. Yeah.
Because a specific IgE will often skyrocket, you know.
Particularly in the very young. Yes.
Exactly.
Yeah. In
It's
infants.
different
Yeah.
ballgame.
Infants, young toddlers. Yeah. Yeah. We're studying dupilumab with milk allergen oral immunotherapy, and our main goal is to understand if gastrointestinal side effects of milk oral immunotherapy are improved with adjunct dupilumab. And this is a study that we're doing in collaboration with Phoenix and Mayo Children's in Arizona with Ben Wright and Matt Rank. The other hot biologics or small molecules that I think are really interesting are BTK inhibitors. And so this is Bruton tyrosine kinase inhibitors, and these are important for B cell signaling and B cell maturation. Also important in basophil and mast cell degranulation activation. And there was a nice small phase 2 study looking at acalabrutinib, a BTK inhibitor in 10 peanut allergic adults. And this was published in 2023 in JCI. After just 4 doses, it's a medication that's given twice a day orally. They assessed threshold sensitivity. So these 10 adults underwent screening food challenges to peanut or placebo, were randomized to receive BTK or placebo for 2 days, and then tested with food challenges. And you can see this amazing, amazing effect of peanut allergic individuals. So this is the max tolerated peanut protein dose, less than 100 milligrams at baseline. And then after 4 doses of 2 days of therapy, the threshold, most of the individuals were able to tolerate 4,000 milligrams of peanut protein, a tablespoon of peanut butter. This is OFC doses of peanut protein. So looking at modified Practol dosing schedule and symptom scores between acalabrutinib in yellow and baseline a placebo in blue. And you can see that very low or few symptoms.
Can you explain the mechanism? I don't understand what's happening.
So it's stopping signaling within the mast cell and the basophil, and inhibiting degranulation and activation of those cells. And so it's pretty amazing to see this treatment effect. I mean, it's only 10 allergic adults. And so I think that there's a lot of excitement about the possibility of this because of its quick onset of action.
Is there a placebo on?
There was a placebo.
And it's allergen agnostic.
Yep.
So-
Yeah. And so, we're reducing, they went on to show that it can reduce peanut basophil activation test, but it doesn't alter specific IgE because it's inhibiting activation within the mast cell and the basophil specifically.
This may be a stupid question, but if you have BTK, you're born with BTK abnormality, you have immune deficiency disease. So if you inhibit it forever, don't you acquire an immune deficiency disease? Can you just do this?
Well, so I think that it is currently approved for leukemias and malignancies. But that is the important concern of using this in the allergic disease space, is that if you're eventually affecting B cells, what will be your vaccine immune response or infectious disease immune response? And so I think people are, the companies that are in this space are really trying to figure out dosing and trying to understand vaccine schedules. I mean, I think this is going to be a very important consideration for taking this from adults into pediatrics. But exciting to think about something that has a quick onset and maybe has a short-term use indication. It was well tolerated, no symptoms while taking acalabrutinib, no treatment-related serious adverse events. And then I know we're short on time, but the last therapeutic that I wanted to touch on is peptide immunotherapy for treatment for food allergy. And I think that there's some really nice phase 1 studies in this space. This is looking at PVX 108 based from an Australian company. And stage 1 was single ascending doses looking for the right dose. Stage 2 was repeated dose administration of 6 doses over 16 weeks. This is a peptide immunotherapy that has 7 epitopes, T cell epitopes for the ARA-H1 and ARA-H2 peanut epitopes. And so it's a combination therapy. And the idea is that this injection is like introducing peanut peptides to the immune system in a carefully measured way to induce T cell changes and skewed towards tolerance. They showed that it's relatively safe in stage 1, looking at all of the different dosing strategies. Mostly mild adverse events, some moderate. When they looked at the multiple doses, the 6 doses over a 16-week time period, there were some severe or moderate reactions, more AEs related to the injections, but the severity of the AEs and the number of AEs reduced over time. I think that's important. This peptide immunotherapy does reduce basophil activation. So I just highlighted that these are the controls. This is peanut stimulation of basophils. And then this is in those patients that receive PBX 108. And as we would expect, that this peptide immunotherapy increases CD4 T cells and decreases the Th2A T cells, which are our allergic T cells of interest that we're really hoping to affect. So I think this is also just phase 1 prelim data, and the company is going to be pursuing larger clinical trials, and I think there's a lot of excitement for this. So I will end that there's many other biologics that are out there, and there's these approaches that are allergen agnostic, which is really, I think, important when we're thinking about our patients in mind who have multiple, maybe not just multiple food allergies, but other allergic conditions. And in an era of precision medicine, this is where we're really trying to hit the mark. But it's not for everybody. And some people will want allergen specific therapy. And there hopefully is some movement on that on their horizon with oral immunotherapy that people are doing in practice, but also epicutaneous immunotherapy, sublingual immunotherapy, oral mucosal immunotherapy, the toothpaste. And then people will use these in combination. So hopefully 10 years from now, we'll have many options. And it'll be up to us to do the art of, what do we start first? What do we do next? And I don't think there won't be a specific pathway or paradigm of how we approach this. It will really be driven by our patients and where they are in their life needs. So the word of the day is dermatitis. And then thank you guys so much for being such a wonderful interactive. Thank you for all of the good questions and comments, and for the invitation to speak here with you all.
Hi, this is
questions.
This is Vinod here from community allergist. Can you hear me? Hello?
Yeah, so that's a great question of with Omalizumab approved for food allergies, how will it affect understanding which patients outgrow their food allergies, and how do we test for this? And this is a great question. The only way you're going to be able to test for that is to stop and wait at least 3 months, because it has a long half-life, as we know, to be able to reassess. And this is a question of whether if you only have a milk or an egg allergy, the allergens that are more likely to be outgrown, is that something that you start somebody on therapy with Omalizumab for? And I don't think there's a right answer. But it does highlight the need for better testing. For diagnosis, that can then also be leveraged for monitoring disease responsiveness. There's a lot of different organizations like FAIR and the NIH and different groups that are really trying to think about bringing basophil activation tests to be more clinically available, but that has its limitations as well. So there's definitely a need for better diagnostics.
Dr. Swan is trying to ask a question. Please, and I don't know if this is
Again,
Can you hear me? This is Vinod.
Let's see. Yep, my volume is on,
Hi, can you hear me? This is Vinod
but I
from.
can't. Is the volume? I think the volume is through here. Maybe chat.
Recording in progress. Could you repeat the question, please?
This is Vinod here. Can you hear me now?
Yes.
Oh, hi. Yeah, thank you. This is Vinod, community allergist. I had a few questions, too long to put in the chat. So, we know the IgE levels are certainly not a great marker for either severity or, how the patients do. So, but the dosing of Xolair is dependent on total IgE. So, do you know if there's, again, interquartile differences between, those, who receive the higher dose versus the smaller dose? And could the ones with, lower IgE, but probably more severe allergies could benefit from a higher dose of Xolair? That's question one. Second was practicality, since pre-filled syringe is the only thing, in terms of home, administration by a parent. We know of 2-year-olds getting immunizations, how challenging that is, but trying to do sub-Q with the pre-filled syringe, how practical would it be for a parent to do that at home? Third would be, Xolair and eosinophilic esophagitis, knowing that IgE is not necessarily the mechanistic way for esophagitis, going on to desensitization. What do we think about, how that's going to affect eosinophilic esophagitis? And last question unrelated to the Dupixent, if any data is there on the birth registry to think about what's the onset of allergies in an infant before they've actually been introduced with foods. Thank you.
you can choose any two to answer 4 questions.
So for the total IgE question, the things, so this is we are trying to analyze all of the outmatched data to really understand disease markers for disease responsiveness. The thing that pops up is the higher total IgE, the higher the chance of clinical success following omalizumab treatment, which is a little bit kind of puzzling as to why that is. But it is something that we see actually even in our phase 2 studies when we look at the data at that 8-week time point and look at, when we introduce foods, we see that pattern there too. And so there was good distribution in all of those dosing ranges. This is a paper to come. This is the data that we're analyzing. So I don't think it's about dosing frequency. But, Philippe Bejean is studying higher doses of omalizumab outside of this dosing table, taking into account responsiveness in therapy with the BOOM study. So I think some of this will be answered with the BOOM study, and that's a omalizumab multi-allergen food study occurring in Canada. Some of your other questions were about EOE. And we don't know. I mean, EOE with food allergy is a very interesting problem. I think we're just beginning to understand it. It used to be, we've had a lot of patients that we've followed for a long time at our cohort at Stanford, where these patients, when they start oral immunotherapy, have a lot of GI side effects. And is that on the spectrum of EOE? And then, but not everybody gets scoped and biopsied to really prove that they have EOE. When we've done this prospectively in the POISE study, we've seen that at baseline in food allergic patients who are avoiding their culprit food, there is baseline eosinophilia that meets the threshold for EOE, but they don't have strictures or, or furrowing or anything like that. So they're not clinically symptomatic, but they have baseline EOs in their gut. And then with oral immunotherapy, that transiently increases, again, asymptomatic, and then decreases over time with oral immunotherapy. With omalizumab, we still don't understand fully how omalizumab plays with epithelial barrier integrity and the repair of the epithelium and how that might influence EOE. And so I think this is a really important question for us to try and understand is we don't really fully understand EOE and OIT, and how does an anti-IG affect that? In clinical practice, Tina Cinder, one of our faculty at Stanford, actually runs a multidisciplinary GI allergy clinic for patients who are on oral immunotherapy in the community and then develop EOE or have EOE-like symptoms. And they're anecdotally treating patients with dupilumab. So, because our food allergy patients, once they start oral immunotherapy, they do not want to stop. And they want to do anything that they can to keep OIT in their diet to protect themselves from IgE-mediated life-threatening food allergies. Now with omalizumab, maybe that's a game changer, but oftentimes we were reducing doses, adding other therapies like dupilumab to keep them on the smallest dose to kind of benefit both diseases. It's an important question that I think we really need further studies to investigate.
Well, thank you again. I have to end this because we
Yep.
have a case presentation for one of our fellows and other conferences to follow. But thank you again so much.
Thank you.